Hi, everyone. My name is Mathew. I'm a graduate student at the University of Virginia, and the title of my talk is Single-Cell RNA-seq reveals FUS pressure-dependent enrichment of transfected cell populations and their differential transcriptional responses after BBB opening. So as many of you know, FUS blood-brain barrier disruption enables targeted, non-invasive, and repeatable gene delivery across the BBB. However, significant translation questions remain, namely, which brain-resident cell types get transfected with FUS, how do these proportions change with FUS peak negative pressure, and how does FUS PNP affect the transcriptional responses of the transfected cells? And I'll go over how we address this question here. Mice are treated with one of three conditions, all of which were given an intravenous injection of lipid-shelled microbubbles and M-Ruby bearing plasmid. The control mice were not given any FUS additionally. At 0.2 megapascals, you can see kind of a dimmer opening of the blood-brain barrier, and at 0.4 megapascals, you can see very consistent bright opening of the BBB. However, I will note at this 0.4 megapascal condition, we still don't see any signs of histological damage. We then waited 48 hours and dissociated those treated brains into single-cell suspensions. In the control condition, we don't expect any M-Ruby to make it across the BBB, and so anticipate no M-Ruby transfection. At 0.2 and 0.4 megapascals, we expect some cells to pick up the transgene and start expressing it. To isolate those populations in the FUS-treated conditions, we then performed flow-activated cell sorting. And you can see that we get M-Ruby positive populations on both flow charts. As well as at 0.4 megapascals, we get a greater magnitude of transfection. And so this leaves us, after flow sorting, with our enriched populations that are only expressing the transgene. And then we pooled all of these cells from all three conditions and performed single-cell RNA-seq. The results of that study are summarized on this t-SNE plot here. t-SNE plots are very similar to principal components analysis. In this case, each dot represents a single cell, and the color of each cell can be mapped back to its condition of origin. We then made a conscious effort to focus on these 6 cell types, namely microglia, neurons, oligodendrocytes, astrocytes, pericytes, and endothelial cells. We did identify other cell types, however, we weren't as confident in their expression levels and their association with the neurovascular unit. We then quantified the proportions of these cell types and compared them to the control condition. You can see at 0.2 megapascals, we have an enrichment for endothelial cells, pericytes, and astrocytes relative to the control condition. And only at 0.4 megapascals do we start seeing more significant expression of microglia, oligodendrocytes, and neurons. This led us to generate the following model, where the likelihood of a cell type being transfected is proportional to the peak negative pressure, but only at 0.2 megapascals do we get an enrichment of cell types associated with the blood-brain barrier, again, such as pericytes, endothelium, and astrocytes. At the higher pressures do we start involving microglia, oligodendrocytes, and neurons. We performed differential gene expression for all cell types, and the expression levels were widely variable on a per cell type basis. But I did want to highlight some genes that were consistently expressed independent of cell type and dependent on pressure. These 4 plots are violin plots of gene expression, and I wanted to draw your attention to the 0.4 megapascal condition. You can see for each gene that's represented by each panel, the highest expression level for all cell types involved is in the 0.4 megapascal condition, with that expression level being significantly attenuated at 0.2 megapascals or the control condition. And if we look at the identities of these 4 genes, we can see that all 4 are associated with cellular stress or inflammation, suggesting that at the higher pressure, even at 48 hours, we are seeing either an active or resolving inflammatory response. To conclude, FUS transfected cell type distributions depend on peak negative pressure, with 0.2 megapascals leading to overall weaker transfection, but a preferential BBB-associated cell type distribution. At 0.4 megapascals, these trends are reversed with stronger, more global transfection. The extent of differential gene expression does depend on the cell type. Unfortunately, we weren't able to show the data. However, we did identify some genes that were associated with cell stress and inflammation that are expressed independent of cell type, but dependent on FUS peak negative pressure. And with that, I'd like to thank everyone in the Price Lab, our collaborators, both at UVA and at Johns Hopkins, and our funding sources. I'm happy to take any questions. Thank you.